Marinda Mortlock, who's actually going to give the talk today. So she's a postdoctoral researcher in the Centre for Viral Zoonoses at the University of Pretoria. And her research focuses on bio and serosurveillance of bat-borne paramyxoviruses at the human-wildlife-livestock interface and assessing risk of virus transmission and spillover. And she's been with the Biosurveillance and Ecology of Emerging Zoonoses research group since the start of her postgraduate studies. And then when we get to the question section, she's going to be joined by Marike Geldenhuys. And Marike is also a postdoctoral researcher with the Biosurveillance and Ecology of Emerging Zoonoses research group. And her focus is on potentially zoonotic coronavirus research in bats. So with that, I will hand over to our guest speakers to kick us off.
Thank you, Helen, for the introduction. So I thought the information that you sent me regarding what you want us to discuss, I thought the title for this talk that would be suitable is Beyond Fear, Educating the Public on Bats, Diseases and Coexistence. So the introduction, Helen already mentioned where I fit in, but I just want to mention that the research leader of our research group is Prof. Wondermark Otter, that has been working on bats and diseases for many years now. So our research group, what we call the bees research group, looks at various different things in terms of bat and small mammal zoonoses. We consider surveillance, diagnostic development. We look at the ecology of these animals, host pathogen interactions, as well as what happens at the human-wildlife interface and interactions that take place there. We also recently established the South African Bat Research Network. I did provide a link there. You're more than welcome to go read up on what we want to aim with this Bat Research Network. So getting into the talk. So to put bat and diseases into perspective, I think we need to take a few steps back and consider human emerging infectious diseases, or EIDs, in general. So it is estimated that about 60% of human EIDs originate from animals. This can be companion animals, livestock, or wildlife. So diseases are transmitted from animals to humans, that are transmitted from animals to humans are referred to as zoonoses or zoonotic diseases. And they can be caused by a range of pathogens that includes bacteria, fungi, viruses, protozoa, etc. So when considering the EIDs of animal origin, about 75% of these specifically originate from wildlife species. So if we look at bats specifically as a wildlife reservoir for diseases, the most prominent pathogens that have been associated with the viruses with them includes viruses. And they have been proven to be the reservoir for several zoonotic viruses. So with reservoir, we mean a species that can harbor a pathogen without any ill effects on them, and then serve as a source for this pathogen to spill over to other species. There can be different steps or a flow to events when we consider the spread of zoonotic pathogens. So it can either be directly from the reservoir host into humans, or there can be the involvement of an intermediate host species. So this can be either other wildlife species, livestock, companion animals, from which the virus then moves from the bat or the wildlife host into these species. It gets amplified to high viral titers, and then it spills over into humans. So once the disease spills over into the human population, it can either result in an instance where there's no onward transmission of the disease from the affected individual. And I've given you some examples of the viruses that we will get into a bit more later in the talk. And then the other option, or the other instance, is where this virus spills over into the human population, and then we see human-to-human transmission taking place. So this can result in an outbreak situation that can either be epidemic or result in a pandemic. There are several conditions that we have to consider that are required for a pathogen to actually spill over to humans. But they all need to align at the right time to be able to cause the disease in humans. So I'm just going to highlight a few of them to put everything into perspective for you. So the host is the reservoir host in the region. So what is the distribution of the reservoir species? Then the second would be the host density. Larger population sizes can affect the transmission and the maintenance of a virus within the specific population. Then also pathogen prevalence. Is the specific pathogen or virus present within the host that we're looking at? Because not all bats within a single colony is necessarily infected with that specific virus, as only a certain percentage of the individuals might be affected. Or even there can be differences between different colonies from different regions. Then there's pathogen release from the host. So is the pathogen being excreted into the environment where it can get into contact with other species, or do we need physical contact with the bat when we get exposed? And then can it survive in the environment? So even though it gets excreted, it might not survive long enough in the environment to actually infect other species and then spread onwards. Then humans need to be exposed. So before they can get infected, they actually need to be exposed. So if you can think about all of these things, they have to be there at the same time, aligning to be able for it to spread over into humans. So what I will not go into detail about is there's several within host barriers. And with that, we mean your immune system. Is the virus capable of infecting human cells and things? So that is already additional barriers that prevents the diseases actually from affecting humans. So we might be constantly exposed to viruses, but they might not be able to infect us. So I think key points that are necessary for the public to understand when we talk to them about bats and diseases would be they need to understand what is the reservoir species, what is the distribution of the species that we say harbors these viruses. Are they present in the location where they are? Are they at risk? Then which pathogens are present? I mean, in certain regions, we see maybe certain pathogens occurring in certain species that do not occur in other regions. And then how are these diseases or these viruses or pathogens transmitted? And then actually which human activities can contribute to the contact and the rate of exposure that they might see for these diseases. So I will briefly go into these 4 points that I think is important for public understanding. If we look at bats as host species, they are extremely genetically diverse, as I think you would have known through some of your other talks. But specifically in Africa, there's more than 230 bat species. So researchers have questioned why bats are able to carry so many diseases or diverse viruses, and which they think might contribute to them being so successful as reservoir species. So they have hypothesized that high population densities, the roosting behavior of bats, their ability to enter torpor and hibernation, all contribute to their ability to successfully maintain these viruses. So additionally, their ability to fly also then allows them to cover vast distances and geographical areas, allowing them to come into more contact with a range of susceptible species that could potentially be infected. Then getting on to the pathogens that are associated with bats. So there was a paper in 2020. So this information is a bit outdated, but I think there is, to prove my point, I will get back to this, but I think after this, there's even more that has been described. But from the decades of surveying bats, where we look at the diversity of viruses that they harbor, at least 29 viral families have been associated with bats. So in totality, by 2020, more than 10,000 virus sequences, which relates to hundreds and even thousands of different viruses, have been detected. And of the 29 viral families and all of these different viruses that we are detecting, there's only 3 families that have actually been associated with direct disease transmission from bats to humans. And within these families, it's only a number of viruses that have specifically been linked to disease emergence in humans. So my point with the data is outdated, and there's a lot more that have come out since 2020, just shows you the vast amount of viruses that we detect that are associated with bats, and only a handful of them actually have spilled over and caused diseases in humans. So there are 3 families that have been directly associated with disease spill over to humans, include your filoviruses, your raptorviruses, and your paramyxoviruses. But I think it's also important to mention the coronaviruses, as although there's a direct spillover link between bats and humans have not been made, we have detected, or there has been several viral sequences that are highly similar to some of the human diseases, human coronaviruses that have been detected in bats. And I think they also warrant some attention. So we believe that bats are asymptomatic carriers because they carry all these viruses, but we don't see any visible signs of diseases. And thus we assume they're asymptomatic carriers. From research over the past few years, considering the immune system of bats, they've seen that the bat immune system is quite unique, as it allows them to suppress viral infection more effectively when compared to other mammalian species. However, lyssaviruses, which belong to the Raptorviridae family I mentioned earlier, or more specifically the rabies and rabies-related viruses, can make bats visibly ill, as these viruses affect the brain and may result in unusual behavior in the bat. So if a bat is encountered that is displaying any unusual behavior, there's a possibility that it could be a sick individual and should be avoided. So some of the things that we can look at, especially for bats that are affected with a neurological virus, such as rabies or rabies-related lyssaviruses, could be behavioral changes. We all know that bats are nocturnal. So they move around at night and they fly. So if we see instances where these bats might be active during the day, it's unusual. If they just hang on a tree or on a wall or something, and they have the inability to fly, it's a red flag. Then you also have increased aggression that might be observed, paralysis, disorientation, and even vocalization. So they can express different behaviors that might indicate to you that the individual is sick and the bat should be avoided. If we think of routes of transmission of diseases in general, or in other words, the way by which a pathogen is spread from a reservoir to a human or other animals, it can be either through direct contact. This would be physical contact with an infected animal, either through touching, scratching, or bites that take place. It could also be instances where people would hunt the animals, they get into contact with the blood and the organs of the bat directly. Then the other option would be indirect contact. And with this, we mean contact with animal excreta, like urine and fecal, either directly with urine and fecal or through environments that have been contaminated with animal excreta. So a lot of viruses are excreted through the urine and fecal of bats, and that creates this contact point where there could be an exposure. So then another option is airborne or droplet transmission. So this is when the pathogen is spread through respiratory droplets and can infect people nearby and remain in the air over for long times and over long distances. Then lastly, it could be through disease transmission, could be through vector-borne transmission routes. So this occurs when an infectious agent is transferred by an insect vector. So if we consider our knowledge that we have for the currently known zoonotic bat viruses, direct and indirect contact seems to be the most prominent pathway through which these diseases are transmitted. So I'm just going to do a quick Q&A, and Marike will assist me with this, where I want to know what are prominent direct or indirect contact points between humans and bats. It can be general, or if there's something specific to your country that you think does not necessarily or is not necessarily generalized across Africa or something, what do you think are prominent interfaces? You can either raise your hand or give an answer in the chat. I'll give a few minutes for that. So anyone that wants to give it a go? Someone said grounded bats and the person finding them. Yes. Bats in roof, definitely. I think that is very common for bats to be or a common contact interface between humans and bats. Bats in roofs. Any other suggestions? Contact with fecal matter, yes. Okay, so we've gotten a few suggestions from the audience. So I also listed a few contact points that I think is important for consideration. So some examples for direct contact would be bat hunting, butchering, and consumption. Bats that enter houses where people actually have to catch the bats and actually remove them from the house, so there is that direct contact between them. Or something we don't regularly think about, but rehabilitation of sick and injured bats, because there's many rehabilitation centers that would take in bats that are injured or sick that people actually hand into them. So then the people catching the bats and the rehabilitators actually have that contact with the bats. Then if we think of indirect contact, some examples would be contaminated fruit dropped to the ground. So as you know, a lot of bats are frugivorous, so they eat fruit, they spit out the pulp, they throw down half-eaten fruit, and people can get in contact, and even livestock or other wildlife that's in the area get into contact with those contaminated fruits that are that could possibly contain infectious virus particles. Another form of indirect contact would be in your attics and roofs, as was mentioned by one of the participants. And we know that in some instances, we even see urine and fecal drop down from crevices in the ceiling that goes into the houses or on the outside of houses. So there is that exposure to urine and fecal matter through that situation. And then another indirect contact through urine and fecal would also be entering caves, whether it's for guano mining, tourism, or anything in that sense. When entering the cave, you are underneath these bats, they defecate, they urinate on top of you, and you get exposed. So based on this, there are several human activities and exposure interfaces that can contribute to disease transmission. And these can be broadly grouped into 4 categories. So the first would be shared food sources. So my example that I put here for you is fruit trees that are planted in yards or even orchards where fruit bats would go in, eat the fruit. You have these potentially contaminated fruit dropping to the ground, getting picked. Sometimes people don't wash fruit when they eat it beforehand, and there's that exposure at that point. Then in terms of common environments, we have things like human-driven landscape or environmental changes, such as deforestation or shared living spaces, like we mentioned, bats moving into houses, in the roofs due to urbanization. Then there's also animal products or consumption. And I think a prominent example here would be wildlife hunting. Not necessarily only bats, but even other wildlife species that could be potentially infected and through an intermediate pathway infect humans. And then lastly, occupational exposure. As I mentioned, rehabilitators also get into contact. Researchers that work with bats that can come into contact, whether direct or indirect. Some important emerging infectious diseases that have been directly associated with bats include viruses such as Ebola and Marburg filoviruses, Nipah, Hendra, and Susuga paramyxoviruses, and then lesser viruses such as virus. Additionally, as previously mentioned, known human coronaviruses such as SARS coronavirus 1 and 2, as well as Middle Eastern respiratory coronavirus, is also important as there has been related viruses detected in different bat species, though direct spillover has not yet been proven. So to zone in specifically on what is important from an African perspective, I will go into a bit more detail for the paramyxos, the lesser viruses, and the filovirus. So lesser viruses in the Rhabdoviridae family, referring to rabies and rabies-related viruses. I'm sure you all are very familiar with rabies virus, but I will still give a brief overview of rabies. So it's a neurological or a neurotropic virus, which means it infects the brain, and it primarily spreads through saliva of infected animals. So you would get contact through, typically through a bite of an animal. So a common host that we usually associate with rabies is dogs, raccoons, skunks, foxes, and then also bats for certain parts of the world. So infection with rabies, the symptoms involved when you're infected with rabies is usually generalized at first and could be mistaken for influenza-like disease, but it later progresses on with more neurological symptoms. Prevention of rabies is through vaccination. It is a vaccine-preventable disease. And pre-exposure vaccination is recommended for high-risk groups, such as your veterinarians or animal handlers. So for treatment, immediate cleansing of the wound needs to be done, and then that should be followed up with post-exposure prophylaxis within a 72-hour window for it to take effect. And this involves a series of vaccination to prevent the virus from causing disease. So once symptoms actually appear in an infected individual, rabies is almost 100% fatal. So early treatment after exposure is very important. In terms of rabies in bat, so this is quite prominent in the Americas, and they've reported that around 22% of the bat species that occur in Latin America are confirmed hosts of rabies virus. So where does that put Africa in terms of bat-associated lesser viruses? So we do not have rabies virus in the African bats, but we have what is called the rabies-related viruses. So they belong to the same genus as rabies virus, the lesser virus genus, but they are different species. So there are 5 lesser virus species that have been detected in bats from Africa. This includes Duvenage, Shimoni, Matlu, and Falabat lesser virus. Of these, it's only Duvenage lesser virus that has been associated with diseases in humans. And the clinical manifestation of this is in line with what you would see for rabies virus. So Duvenage virus has had, or there were reports of 2 human infections of Duvenage virus, and these were 36 years apart. So as you can see, this is not something that occurs frequently. So the first case was in the 1970s, and it was a fatal human case that resulted from a bat bite. In the years to follow, some bat species were linked to Duvenage virus. The first was Miniopterus, but that is presumed based on circumstantial evidence. And then the virus was subsequently detected in Nycteris sabaica. It was only in 2006 that the second fatal case of Duvenage virus was reported. This was a 77-year-old male where a bat flew into his room, and it was reported that the bat didn't bite the individual, but it landed on his face, on his glasses, and as he was trying to get rid of the bat, it scratched his face. So he did not follow this up with post-exposure prophylaxis, and he died 14 days after the onset of symptoms. So the other viruses, Shimoni bat virus, Matla Matlu and Fala bat virus have not been associated with diseases. And these viruses are associated with Macronectaries vittatus for Shimoni, Miniopterus natalensis for Matla virus. And this virus was actually detected during some of our routine surveillance in South Africa. And it's most closely related to West Caucasian bat Lissa virus from Asia. Then Fala bat virus was detected in Nyctocyanops schleienfi, if I'm pronouncing that correctly. And this bat was submitted to a rehabilitation center as it was displaying neurological signs, and it died after 24 hours. And this bat was then sent to us for Lissa virus testing, and that's when the new Fala bat Lissa virus was detected from this individual. So the last rabies-related virus is Lagos bat virus. So it has been detected more frequently than the other rabies-related Lissa viruses in Africa, but it's also sporadically and not in high numbers. And this virus has been linked to Rosettus, Eidolon, and Ecmophorus, so that is fruit bat species. And all of these have a vast geographical distribution across Africa. So detections has been reported from several African countries, including South Africa, Central African Republic, and Ghana. And this just shows the wide distribution of Lagos bat virus across Africa. So just of note, so although this virus has not been associated with human diseases, it has spilled over into companion animals such as cats and dogs. So if we know what the bat species, which bat species is associated with a specific virus, we can make some inferences of the risk of this virus across the geographic distribution of the specific species. And this is why it is very important for us to identify the hosts of these viruses. So given the route of transmission that Lissa viruses is through direct contact via bites or scratches, I'm asking the audience again, what should you do when you get bitten or scratched by a bat? You can again just leave a message in the chat, or if you want to answer, you're more than welcome. Anyone want to take a go?
I'll go.
Okay.
So if you get bitten or scratched by a bat, you should wash the area with soap and water, let it bleed for about 15 minutes, if I'm remembering correctly. And then you should go to your nearest hospital for the post-exposure shots. At least in South Africa, we're able to do that and say that we've been exposed and we need our post-exposure jabs there.
Yes. So I agree with what you're saying. So we recommend the same procedure as you would follow for a dog bite when you expect a rabies exposure. So as you mentioned, wash the wound with soap and water and treat the wound with disinfectant. Then something I just want to highlight, and maybe this is from a disease research perspective, is if you can identify the bat. We're not asking people to try and catch the bat while you're bleeding from a bite or anything, but if you can identify anything about the size, the color, or distinguishing markings on the bat, that is very helpful for research purposes and to be able to identify what host was actually associated if this should result in a situation where there was exposure to rabies. Then lastly, the post-exposure prophylaxis. Okay, so then for paramyxoviruses, I'm going to start with Saturga virus, because this is actually the only paramyxovirus that has been directly linked to a spillover from bats in Africa. So it was a case where there was a field biologist that was on an expedition for 6 weeks in Uganda, and the individual fell ill after returning back to the United States. And they were able to determine that the etiological agent that was causing this disease was a new paramyxovirus that is related to another virus that was detected through surveillance in bats. So that's where they made this link that that the possible reservoir could be bat species. They went on to go using a specific test for that specific virus and testing the bats and animals that she came into contact with. And they were able to determine that the likely spillover event took place from Rosetta egyptiacus into this individual, but the route of transmission was not known. So this was a non-fatal case. The individual recovered after 2 weeks and was able to go home. So this is the only single human case of spillover of Susuga virus into the human population. There has been follow-up studies specifically looking at the specific virus, looking at which tissues it infects, how it's excreted, and it was found that it is excreted in urine, but it's more prominently associated with the gastrointestinal tract of the individuals, and it's hypothesized that it was through contact with contaminated fecal material that the individual got sick. So this was from Uganda, and we have also initiated paramyxovirus surveillance in South Africa. We've tested a lot of samples from the same bat species, Rosettas, that has a wide distribution across Africa, and we have not been able to detect it. But a recent paper that was published last week, I think, where they also showed high detection rates of this Susuga virus in bats from Sierra Leone. What we saw through our paramyxovirus surveillance was the detection of a lot of related viruses that are similar to the Pararubula genus, to which Susuga virus belongs, but none of them are highly similar to Susuga virus. What we've also shown that is present in the bat species in South Africa, specifically Rosettas, is we detected a virus that is highly similar to mumps virus and human parainfluenza virus 2. So we are still doing follow-up research on this, and the zoonotic potential of these viruses are not known. So we don't know yet if it's capable of spilling over. Has there been instances where the virus actually did cross over to humans, causing disease, but it gets misdiagnosed as something else, or it's underreported? But a study conducted on another mumps-related virus, also detected in a fruit bat up in Africa, but this was from Epimetheus, was able to show that that virus is capable of being infecting human cells, and the vaccine, the mumps vaccine, actually would protect against this virus. I'm going to quickly talk about Hendra and Nipah virus. That's also part of the paramyxoviruses. So I know this is not Africa-specific examples, but it's very important to understand the context for Africa. So Hendra virus emerged in 1994. It caused a lethal respiratory disease in horses, and this eventually spilled over from the horses, which are the intermediate host, into humans. And the virus has a high mortality rate of 57%. So this virus is actually a very good example where you have transmission through an indirect bat contact involving an intermediate host species. So during the outbreak or the outbreaks or transmission of Hendra virus to humans, it was due to horses being kept on farms where there's fruiting trees. And these Theropus bats would come, feed on the fruit, and the fruits would drop down, be contaminated with Hendra virus. The horses will consume them, and then the veterinarians or owners getting into contact with the horses actually contracted the disease. The second example is Nipah virus. This emerged a few years later, also in the 1990s, and caused severe febrile encephalitis in humans. It had a high case fatality rate in the initial emergence of 38%, but the initial emergence of Nipah virus was similar to Hendra virus, where it made use of an intermediate host species. In this case, however, it was pigs. And then it subsequently spilled over to farm workers and farmers getting into contact with these infected pigs. This unfortunately led to the culling of a lot of pigs and actually had a huge economic impact in Malaysia due to the emergence of Nipah virus. So this is a good example of an indirect route of transmission directly from a bat source, as well as through an intermediate host with additional transmission events within the human population. So I say that because during the second, there was another outbreak that followed in Bangladesh. So the first was in Malaysia, and this one was in Bangladesh. And during this emergence or this outbreak, they were able to see that there's actual direct bat to human transmission, even though it was through an indirect route. So as you can see in the little picture at the bottom, it's a bat roosting in these date palm trees. And in Bangladesh, they would collect date palm sap and they would drink it raw. And the bats would also go into the tree, also drinking from the date palm sap, and they would defecate and urinate into these bowls, and the people would consume it and they get infected. Then there seems to be a southward spread of Nipah virus outbreaks that is occurring, because more recently there was reports of Nipah virus outbreak in India. But for this specific case, it was assumed that the individual just got into contact with bats roosting in a well. So if we think about the three different scenarios here, or the outbreaks of this virus, it is important to when you assess the risk of viruses, Nipah virus or any other virus, it's important to look at the conditions regionally or locally, as they may vary across places, because I mean, not everywhere do people collect date palm sap. And I just want to point out that on the top picture on the right-hand side, you'll see an initiative where they covered the date palm sap with reeds to prevent the bats from sitting there and drinking the date palm sap. And that has actually led to a decrease in the amount of Nipah virus spillover events that took place. Why is this important for the African context? So through biosurveillance studies considering paramyxoviruses in African bat species, there has been a large diversity of paramyxoviruses that have been described. Some of them are closely related to Hendra and Nipah virus. So they belong to the same genus, but a different, but there are different species. And as you can see, the species that have been associated is fruit bat species, predominantly, or only fruit bat species, actually. And they have quite a vast distribution across Africa. So one study was that considered human exposure was actually able to show that 4% of the human samples that they tested, serum samples collected from blood in Cameroon, were positive for Nipah viruses. So they used Hendra and Nipah virus, but in Africa, we don't have those specific species, but we've got related viruses. So we do see that people do get exposed to these viruses, but no diseases were necessarily reported as a result of this. But they were able to conclude that some risk factors for exposure to these Nipah viruses included contact with bats, either through butchering or hunting the bats, and then also to some extent deforestation. Then lastly, looking at the filoviruses. So they belong to the family Filoviridae, as I mentioned previously, and this family includes some of the most pathogenic viruses that are known to affect humans. And I think the two most notable genera within this family are the Ebola viruses and the Marburg viruses, both known to cause severe hemorrhagic fevers. So I just listed here for you some of the outbreaks. And as you can see, there's been quite a few outbreaks of these viruses in Africa with high case fatality rates that can range up to 100% for Marburg virus and even 88% for Ebola viruses. If you recall, during 2013 to 2016, there was the largest outbreak of Ebola virus reported in Africa, affecting more than 28 individuals and resulting in more than 11,000 deaths. And this outbreak was widespread throughout Sierra Leone, Liberia, and Guinea. Just quickly on Ebola virus. So the reservoir for Ebola virus is actually still unknown. There has been links made with certain bat species, including Epomophorus, Hypsignathus, and Miniopterus, but whether they are the actual reservoir still remains a question, as there's been no virus that has actually been isolated from the bats. So Ebola virus is mainly spread from primates or other wildlife to humans. And then it's believed that bats form part of this transmission cycle in some way, but we still need more evidence to confirm that. The Bombali virus is actually a new Ebola virus that has more recently been detected. It has not been associated with diseases. I'm just going to mention this because it's been associated with molluscid bat species, and these species do tend to roost in the roofs of houses, but they expect low pathogenicity in humans through some of the research that they have done. For Marburg virus, the reservoir has been determined to be Rosetta aegyptiacus, and it, as I mentioned, it has a broad distribution across Africa. Research has shown that when there's an increase in young individuals in the population, when there's a birthing pulse, it seems to amplify the viral concentrations within the population, and you would see these peaks of virus detections that can be linked to outbreak situations. So up in Africa, Rosetta aegyptiacus has 2 birthing pulses. And that's why they almost say that the amplification of the virus within the colonies is almost predictable on a biannual basis, as this is linked to these young individuals that enter into the population. Marburg virus has been detected in some opportunistic species, like Miniopterus or Hepatidrus. So I just want to point out this, because this is very important for the context of public understanding. So due to uninformed human intervention that failed, that was fear-driven, it resulted in another outbreak of Marburg virus. So it was in Kitaka mine, where there was an outbreak of Marburg virus in 2008, 2009. There was this large colony of Marburg virus that resided in the cave, and due to a fear response from the locals, they covered up the cave. Masses of bats were caught and culled. And in 2020, there was again an outbreak in Uganda. And when researchers went back to look at the cave, they found that the roost was colonized. Even though it was a much smaller colony of bats, this stress that was placed on this population resulted in higher seropositivity or higher positivity of the bats. So more individuals compared to what was previously seen were positive for Marburg virus in this community. So human actions actually driven or drove stress or the virus to amplify more and resulting in another outbreak. Briefly, just coronaviruses, like I said, not directly linked with bats, but we do have a distribution of diverse coronaviruses that have been detected from bat species in Africa. Some are SARS-related, found in your Rhinolophus bat species, and MERS-related, found in your Vespa bat species. Briefly, through some of our surveillance, we've been able to show that there's certain periods where we see these viruses, like coronas on the left and paramyxoviruses on the right, where they excrete at higher rates.
Ek kan verstaan dat ek nou op my foon is. Kom hier so, ek vir jou liefie. Kom net 'n bietjie liefie. Maar jy met jou
I'll continue. So there's certain times of the year where the viruses are excreted at higher amounts, and those periods we can then inform to the public that your risk of exposure is much higher during these periods. So that is very important to look at biosurveillance or surveillance data in bats for diseases over a longer period to be able to pinpoint these time periods where the risk for humans will be much higher. So then lastly, how to communicate with the public. So I just have a few points that I think is important to consider when talking to people. So the first would be risk-benefit messaging. It's important to communicate both the risks associated with bats and diseases, as well as benefits that bats actually have to the ecosystem to provide a balanced view. And I'm sure you all are familiar with the ecological importance of bats as pest control in terms of pollination, as well as seed dispersal. So maybe when communicating to the public, you can use a statement like, while it's true that bats carry diseases like rabies, and in some regions, viruses that are similar to the ones that cause serious disease in humans, the risk of transmission to people is extremely low when proper precautions are taken. It's important to provide clarity, ensure that the message that you give to the public is clear and it's easily understood. Avoid the use of terms that they will not understand, that will confuse them. So I just gave a few examples there. So instead of saying, oh, zoonotic disease, you say disease in animals that can pass to humans. Or reservoir, it's an animal species that carries the disease. And it's also important to use examples or analogies that are relevant to your audience and to avoid assuming that they would understand complex situations. It's also important to manage uncertainty, to be transparent about the level of risk and the uncertainties associated with bat-related diseases, but provide context that can help mitigate unnecessary fear and panic. So just another statement that you could possibly use is most bat-related diseases like rabies or rabies-related viruses are very rare. And the likelihood of transmission to humans is extremely low if you avoid handling bats. However, there's always a degree of uncertainty when it comes to wildlife and disease spread, as new diseases can emerge. So we can admire them from afar, but avoid disturbing their habitats and stay informed with health recommendations from local wildlife experts and public health authorities. Direct behavior. So promote specific behaviors that you advise them to do to help contribute to disease prevention efforts. Like avoid touching a bat, wear gloves, wash fruit before you consume them. If there is feces in your home, clean it with bleach, because bleach is a detergent that you can use that will actually kill the viruses. Then cultural sensitivity. You must communicate or you must tailor your communication strategies to be culturally sensitive for the communities being addressed. So we can't go to communities that are dependent on wildlife hunting and tell them, no, you should stop. You cannot hunt bats anymore. Rather guide them towards safer practices when handling bats, such as wearing protective gloves while performing these activities. Lastly, visual aids. This is a very nice booklet that was put together as an initiative from the USAID Predict project. That is written in layman terms to guide people to understand how they can safely live with bats. And we've recently also made posters that we use to help the public understand these things. So just briefly, the booklet, as you can see, visual representation, what must you do, basic language so that they can understand how to live with them, what to do if something happens when they're in your house. So this is actually a very nice document to go look at. And I provided the link in the presentation if you want to follow up on this. Just lastly, a few key points the end message that I wanted to make of this talk is that a significant number of pathogens, including viruses, are continuously being detected or linked to bats. But only a small amount, a handful, has managed to spill over and cause diseases in humans. So if bat displays unusual behaviors, like flying during the day, they might be sick, avoid contact. And urine and fecal are prominent routes through which the viruses are excreted, and direct contact with urine or fecal or in environments that could be contaminated with bat excreta should be avoided or cleaned properly and precautions taken. Lastly, understanding and respecting bats is crucial for coexisting harmoniously with them. And we can only achieve that through public education on the importance of bats and instilling measures to coexist safely with them. And with that, I've reached the end of my talk. Thank you.
I had a question, I guess, kind of when you're talking about Marburg's virus, and I know it's been kind of detected in rosettes, but I've never heard of a case where it's actually been transmitted from a bat to a human. Is that true?
And maybe Prof MacArthur can answer this a bit better, but I think a lot of it has been circumstantial, where there would be an infection, and then it was linked to the person entering a cave where they know the bat's roost, they know the virus is excreted. Marike, you can correct me if I'm wrong, but yes.
Yeah, so it's hard to prove this bat flew into my face, it had Marburg, and now I
Unless
have
you
Marburg.
catch it. Yeah.
Yeah, right? Which that doesn't happen. So what I do know is that, for example, miners that go into the caves, some of them get sick and then there's an outbreak. Then they actually went and investigated the bats in the cave and they found the same strains of Marburg. So I would say that's pretty much definitive.
Pretty strong. Yeah.
They were there, they've got the same virus. There's not a lot of, if any, genetic differences between them. So yeah. You know, direct contact,
direct contact, great. Yeah, no, because it's just, I suppose it's the, you know, if you sometimes hear of people guano mining and they, you know, kind of never get ill, and then you hear of somebody kind of visiting just, you know, in an area and then they get, and you just think, well, actually, you know, so I suppose it's that kind of, but yeah, obviously it makes sense if it is somebody who's been mining in the area, exposed quite a bit, and then there's an outbreak, that makes sense. So Lorraine, you've got your hand up.
Yes, very interesting talk. Thank you. I've got 3 questions. I'm going to just shout them out and just take it from there. My first question is, what informs the higher risk periods, that graph that you had, and how is that changing with the climate change? Is that temperature related, rainfall related? What is informing that? My second question is, I saw on your map for your paramyxoviruses that Botswana was empty. There was nothing indicated there. Is there a reason for that, or is that an absence of data? And my last question is, just to put this in perspective, with a lot of these viruses, are there other reservoirs, or are a lot of these only associated with bats, or is there some other reservoir? You know, just to kind of put it in perspective. In perspective, those are my questions.
So the risk periods. So the current process of the research was to do biosurveillance, looking what diversity we can find in the bats over a continual period, so a longitudinal period. We did that research over 2 years, 2+ years, where we would take monthly samples, and we would see during certain times of the year, we would see this peak in excretion of the viruses. We then went further to overlay that with the climatic data for the region, as well as the reproductive cycle for rosettes. So that has allowed us to make some inferences about what could possibly be driving these peaks. So for instance, the paramyxoviruses in rosettes from South Africa, which is different from the ones in Uganda in terms of the reproductive cycles as well as the environmental conditions, we saw that during winter, we see this massive peak. So that coincides, for instance, with the colder climate. I mean, the lower quality food availability. They can't feed on the natural figs anymore. They have to look for alternative food sources. So it could be temperature related or climate related factors that play into physiological stress of the individuals resulting in this peak. But we hypothesize this. We have not been able to, we haven't looked at if it's actually climate driven, but we've said that it coincides with that. The same with the reproductive periods. We see the peaks that starts on from May onwards, and that coincides with when you have these naive individuals. When there's a birthing pulse, end of the year, you see this influx of individuals. and they waning of maternal immunity, they get susceptible, and then we see this peak. So currently we can only make hypotheses on what's actually causing these viral excretion peaks, but at least for the local region and for the work that we've done with the coronas and the paramyxoviruses, identifying these time periods of higher excretion, we can at least use that to inform the local population that lives around this cave and tell them, listen, during these time periods, be careful, do not enter the cave, try and avoid bats to limit exposure. So the second was the Botswana. Why was it empty? So the plotting that we used for the distribution mapping was based on the African corruption report that is based on museum vouchers that has been handed in. So I don't think that it's because we didn't put it there. I think it's just limited data availability on the distribution of these species, as the plotting that we used was just based on museum records. And then
how far back do you go with that? Sorry. How far back do you go with your museum records?
I'm not sure, because we used the data from the African corruption report. I think I'm not sure.
Back, yeah, kind of a long time, Lorraine. Yeah.
Yes.
Okay.
Yeah, I think they used
long term.
All the information they could get. Yeah.
I just want to know, is the question about the bat distribution in Botswana or about the presence of paramyxoviruses in Botswana?
That is the last one, the presence.
Okay, so we actually have collaborators at what is it, BUST? It's an international university of technology in Botswana, where they're actually doing some of the first work on coronaviruses and paramyxoviruses in Botswana very, very recently. So there should be a publication sometime this year or maybe even next year. So there really hasn't been a lot of research done in Botswana on these viruses. So hopefully that can be fixed in the future. There's also some more studies that are ongoing now where they're looking at molosset bats in in the roofs of schools, because they have large populations that are there making a mess halfway through the year, and then the bats are gone, and then they come back, make a mess, and it irritates the teachers. So we started this initiative with the University at Botswana to see, maybe let's have a look at what's in the bats. We can reassure the teachers, they don't have to worry if it's about the mess, clean that, but they don't have to worry, or maybe they should worry about some things that are happening in the bats as well.
Yeah.
Very interesting.
Maybe I can just add on that. In terms of, like Marike said, there's not research that has been done really there for us to have results on the diversity. For paramyxoviruses, they, based on the research that has been done, it shows that there might be a co-evolution between bats and the paramyxoviruses that we detect. And I can't recall that there has been any bat species that we've tested or that has been tested across literature where we haven't detected a paramyxovirus. So I assume that it's across species you will detect paramyxoviruses. And in terms of disease association, it has only been fruit bats that have been linked to zoonotic disease transmission. And the viruses that are detected from the insectivores are quite diverse, but they've not been related to anything or any known human pathogens or things like that.
Very interesting. Are there other reservoirs of these diseases, or is it predominantly so I think it might be a bit of a vague question, so don't worry too much about that. It's just to put it into some sort of perspective, within other animal species.
Yeah. I think through surveillance, a lot of species have been limited. So there's not a lot of surveillance in a wide range necessarily of mammals to look at paramyxovirus. But they have detected paramyxoviruses, for instance, in rodents and shrews, as well as other mammalian species or even reptilian species. But as I said, with the data that we have now, it seems like co-evolution. So it will be distinct groups of viruses. So that specific virus that we detect in bats does not necessarily occur in other species, although they have reported some instances where there's cross-species transmission, for instance, between wildlife species such as bats and rodents. I don't know if that answers your question.
Yeah, it does. Thank you very much.
I have another example for that. So, for example, coronaviruses, most of the viruses that we know are bat coronaviruses are actually just bat coronaviruses. MERS is an interesting example. The evolutionarily bat coronaviruses, the MERS viruses originated in bats, but the reservoir for MERS is not bats, it's camels, because camels infect people. So long ago, a camel got the virus from a bat, but now, you know, it's expected to come from camels. But for the filoviruses, there are also no associated intermediate animal reservoirs. It's just bats.
Very interesting. Thank you.
Just to take the opportunity to say, as you can see from the group, obviously working collaboratively across Africa, but particularly in Southern Africa, and as we've got this network, it's just really good for you guys to maybe just to realize that there are opportunities. I know I've talked to Vanda quite a bit about, you know, if we actually found any dead bats as well, to try and actually get those tested. So there are opportunities to collaborate and to help. You know, we want kind of evidence-based to kind of make sure we inform those decisions. And I think as Marinda had very, very well put, you know, we do need to hit these kind of arguments head on and give people informed and accurate information. And that's obviously, you know, we don't work on diseases, but the lab also is looking at very much about the ecology and trying to understand the bats' movements as well. So it's a really dynamic and exciting research group. So I'd highly recommend you guys have a look a bit more about some of the research going on, and we'll be been taking a note of the papers you've mentioned, and we'll certainly share those and the details on the lab as well. But yeah, so Vanda is here as well today. So yeah, it's really it's fantastic to see the work and how much has been happening with the lab, and you know, it's really fantastic to see more collaboration across different kind of research, you know, ecology, but also kind of the disease group, very much the One Health kind of look at it, but also to ensure, you know, we also want to make sure we're keeping people safe, but we don't want to, you know, also, you know, there's very low risk, but obviously if there's something happens, it is, you know, quite extreme sort of occurrence if you know, if people are exposed. So I guess it's trying to make sure that we're kind of keeping people safe, but trying to also ensure it's not alarmist, and then we kind of have these mass culls that then are not good for people or bats. Just wanted to say a huge thank you very, very much to Marinda and also to Marike for coming along with the questions. Marike's also given a talk for us before as well, so which is fantastic. And Vanda will be giving a talk at our Winged Wednesday webinar series later this year, so we'll be sure to share that with all of you as well. So thank you very much.